Attomarker is essentially a blood testing company. What we have developed is a form of point-of-care blood test from a single prick of blood, which can be performed in 5 minutes. Our ultimate objective is a handheld device, which we've already developed. But in the short term, and when we're talking about COVID-19, we're actually talking about a benchtop device, which is available already and being used. There is a chip in the device which has 170 gold nanoparticles printed on it, and which is then functionalized by printing an array of additional elements on it, which in the case of COVID-19 are going to be the recombinant antigens S1 and S2, which detect the spikes on the virus, and the nuclear capsid protein, along with the SARS membrane and envelope proteins. It will also perform a C-reactive protein test, which is effectively a test of inflammation. It effectively tells you that a patient is very ill. What makes Attomarker unique? It is a multiplex testing system. It's possible to do all of these tests simultaneously from the single prick of blood, which allows you much faster assessment. It also means that you don't need to be taking blood in quantity from patients, nor do you need to ship it off to path labs. You can do it in front of the patient at the time. One of the other things that the multiplex testing does, and a lot of what is going on at the moment, is single tests for antibodies. We don't think that's probably adequate. You actually have to be testing for more than one thing at a time to get a proper picture of the disease in the patient and of the degree of protection. This isn't a test for have you got the disease. It's much more important for how do we get everybody back to work. We've now been testing in St. Thomas's Hospital, which incidentally is of course part of King's, in London for the last 4 weeks in conjunction with them. Those tests are going to produce a research paper which is going to be submitted for publication either today or tomorrow. And Thomas itself is now finalizing validation of the tests with a view to putting the device into play in its hospitals as soon as possible. Where we are looking at using the technology immediately is on, first of all, staff surveillance. There is already testing going on, but hospital staff can be placed under daily surveillance, which allows immunity to be followed. It allows the hospital to promote immune staff to frontline work and to test self-isolating staff to ensure whether they have or haven't actually got the disease. It also allows for triaging patients at the door to the hospital to allow the immune patients to be allocated to a green care pathway, and the non-immune patients can then be properly treated with additional care in the COVID-19 wards. It also allows ultimately the authentication of immunity. In the future, we will be able to use these tests to assist in vaccine validation. So in other words, we can test the efficacy of vaccines by measuring in the field degrees of herd immunity in order to suppress the threat from the disease as a whole. We are now in discussions with a manufacturer to industrialize the process of the benchtop device. The chips for use in the device are already being produced at volume in Berlin. Abbott have an antibody test which they can roll out at scale, but it is a single test. It is not a multiplex test. And the research and work with Thomas indicates that you get a more complete picture of what you're looking at if you can test for more than one antibody simultaneously. Those tests are also take rather longer. The complete turnaround for an Attomarker test is 10 minutes. And you can do that alongside the patient, essentially. There are a lot of people working on antibodies at the moment. They just aren't doing it in the same kind of way. We're looking for $7 million US to complete our Series A round, which allows us to develop this at volume, but also to continue with the production of the handheld device and complete the regulatory processes for that.